All right, let's get started with our last week of classes. Let's do this. All right, and so here we are. And so we won't have a discussion, as I mentioned last week. We won't have a discussion today. We'll meet today and Wednesday. And so what we'll do, we don't have more class discussions, we don't have more reading assignment, but we'll have some new material as we try to pull things together. And you're working on your capstone projects, and let me know when you're done with those, and let me know if I can help you along the way. Then our final exam is a week from Wednesday in here. It starts at 11. And so we want to talk about that this week and pull things together for that, okay? And so in starting to think about our final exam, in general, you should strive to describe different examples for the different questions. You'll see I have some suggested questions, and we've talked about suggested questions already. And there is this potential overlap. You know, it says like, oh, you know, pick an endocrine disruptor and describe, you know, such and such. And then the next question is, what about an description that does that, right? And the goal is to try to use different examples like that. And so we'll talk about that, but, you know, the goal is to try to bring as much as you can to the exam. It's important to think ahead of time, you know, not to just have, oh, I have one example that'll work for every question like that, you know. So we'll try to get some different answers and different examples, because we have a bunch of good ones like that. So we'll be thinking about that, talking about that. So here's what I have today. I say, let's talk about some examples of possible questions. And what I imagine the final exam will be is like 10 questions, 9 or 10 questions, and you'll be asked to write on 5 of them that you'll have an option for. And the ones we talked about today are ones that I thought, well, this would be a good question for the exam. And one of them is, do you have any questions that you think should be on the exam? So, you know, you can send me questions for the exam if you think, oh, that'd be a really good one. And so these are ones, you know, I just thought, right, the first exam I sent them out. This exam, I'm not going to send them out, but let's start getting organized because you have to try to write long answers, right? So here's one, and we talked about some of these already. I said, oh, that'd be a good example. This is a good example, right? Whole idea of the sensitive period. So you can use a medicine at a different time if it's not the sensitive period, you know? And so, you know, this recording will be posted so you don't have to scramble to write down the exact writing, but also this is just kind of a draft of a suggestion, but I think it's a good question for the exam, and I talked about it before, right? So drugs that are used but are also endocrine disruptors or teratogens like that, and the reason is because there's a sensitive period. So it's a really great way of underscoring the concept of a sensitive period like that. And so describe them in detail. What problem do they cause? And there's a good chance that you'll want to pick one of these. And I talked about this last week. This is just our first example. Pick one and learn a bunch about it. And learn about, you know, that strategy of, well, what is this molecule bind to? What does it interact with? What's the initial molecular issue, and then what's the pathway that it, you know, is causing trouble with, and then what's the adverse outcome? You know, so it's that adverse outcome pathway. Try to write up about that, and you could pick more than one of these, and maybe you'd use one for a different example and a different question like that. You know, they're good ones. And so we can go over some of these like that, and or go back to them, but that's an example of the first idea like that. And it came up when we talked about Accutane, and it was like, okay, that's a drug, and we got to be careful using it, right, because it causes birth defects. So pregnant women cannot use that Accutane, because it's a retinoic acid mimic. And we learned that it causes apoptosis, both in its natural way of being a good drug for decreasing acne by killing oil-producing cells in the skin, but also apparently that's a big way that it causes trouble during the development of fetus. Unborn fetuses can have some apoptosis occur in really important cells in different organ systems. So great example, and be able to explain that or one of these other ones. So let's just pause with that. And do people have questions about this question, or anything about that? So we'll just look at this question and we can see what you think about it, and if you have questions or issues. And then if you don't have any today, maybe you'll have some on Wednesday, right? So you'll start thinking about this and saying, yeah, even though I talked about this last week as a good question, this is a good question for the exam, and you should be preparing an answer for it. And if it doesn't have to be on the exam, you'll be able to plug that example in somewhere else, like that, okay? So anybody have any questions or issues, thoughts about this? Another thing is you can find another one too. You can say, okay, I'm going to want to learn about this, and I've already found one, or I'm going to find one like that. Okay? All right, so that's an example. And that I just include some of this stuff we already looked at, because you want to just say, okay, I got it, but actually, find good examples, because they have features like this. Oh, this idea that the actual molecule that's initially the exposure molecule, like mercury, is not such a bad molecule, but methyl mercury is a very bad molecule. And so there's a conversion, and sometimes the conversion happens within the organism, like in these drugs. Other times it's another organism in the environment that converts it to a problematic molecule. And so we want to get details like that and decorate our answers with these details of the principles in this course. And you can plan on your studying to organize it in that way. Okay? So just more examples like that from slides we already looked at. The idea that, apoptosis is a big story for this Accutane. Great example like that for that. And then another one that Luke brought up is that the molecular pathway includes FOXO, so we have some stuff that we learned about throughout our course like that, and molecular mechanisms are really great to get to in answers, okay? And Bryce brought up valproic acid, and I shared the notes he had. It looks like another really good example. Now, I brought this example up because you might say, I want to use that example, but I haven't learned as much of it as I should. So you can spend 20, 30 minutes learning more about valproic acid and work on building up toward an answer. Not necessarily an answer that you memorize, but just key things that you would want to know in order to get a good, full answer to that question, okay? All right, and so here's another question. I said before that, most of the questions would be like a broad concept, and then you get to pick the example. This is just such a good example that we talked about, was in our reading, that it should be just a straight out question of like, describe that DES daughter situation and explain the history of it and the problems that it caused, like that, yep. And we really like to dive into the molecular pathway. This is a great example of that. We like to dive into the pathway, especially when it's a developmental biology pathway. We like to do that, or that is a good example of that. And it's a famous example of the Hox genes from developmental biology discovery. So it's just a really great example. And then we also like to set it in environmental context. So this is an example of a medicine, and this medicine was used in the United States. And so we want to have some of that history to set the stage for that. And it would be similar encapsulated in a way similar to the kind of writing you're trying to do for your capstone project, right? To make it a nice balance of setting the stage for it and then diving into it with some details, yeah? So we've talked about this, and I just, included the slides that were some of the slides we used. It's rooted in the development of the vertebrate reproductive tract. The vertebrate reproductive tract has a strong anterior to posterior organization, and there are genes that are really important in all animals that have an anterior posterior axis called Hox genes that are really important setting it up. And so that's the problem with DES, is it causes this homeotic disruption of or transformation of the female reproductive tract, and that leads to a tendency to develop certain types of cancers, and a tendency to not be able to carry a pregnancy to full term, and it's because of the misexpression of these Hox genes, and so just a really good example of that. And so I'll just go back to that and pause and see if anybody wants to go back over that. And you might say, oh, I'm not ready to talk about DES right now, but I'll get ready and talk about it on Wednesday, because I'm saying that's a good example that you want to have good information to write about on the exam. Like that. And so you can see, even if we don't have an example like this, you can see I'm not picking like footnotes. These are things that we read about and we learned about that were pretty, big topics like that, okay? So here's another one, and again, we don't need to hurry about through this because this is our main thing we want to do this week, is talk about the final exam. So, so this one is where you're going to actually write about your capstone project some, by comparing it with another example from our course. And so if your capstone project is an example from your course, well, you have to pick another example from our course and draw comparisons between them, how they're similar and how they're different. And I like that because all of this is really interrelationship between what we've been learning in our course and what you've been learning in your capstone project, like that. And so you should be ready to be kind of an expert in your capstone project. And lots of that, maybe all of that, would not be from our classroom at all, but you can bring that example to our exam. One of the reasons for writing this question is because that's a good example to use on the exam. Even if I didn't explicitly say, hey, what about this? You should be, saying, I'm looking for a way to use my capstone project as an example on this exam because I'm kind of an expert, and that, fine, that's absolutely fine. So here I'm saying, yeah, let's actually use it on an answer, and then not use it on another answer where you're just like, oh, capstone project, nice, like that. And so and think about it. And so here, and this is kind of a double dipping thing too, because you might write about that in your capstone project too. Oh, before I forget about it, I meant to describe this before. So remember, we've done the short writing assignments, and now you're writing the science part of your paper, like that. So you could have your short writing assignments just be, oh, this is a section on my paper. But especially with the ethical issues, if you can find ways of fitting those into the capstone project writing and not just be a separate, oh, this is my 2-page assignment, like that. It's pretty easy to try to put that throughout the paper, bring up these topics that you ran into, like that, and more, use that as a guide to try to help you address those. And then it's harder to say, where do you fit in, like that. But for example would be to write your capstone project about the importance of this research on this topic. And then you fit in partly because why aren't you doing this research, right? Why aren't you moving toward this, or are you moving towards something else that is of significance, right? And it could be in this field, or it could be another field. So I forgot to mention that idea, that in writing those ahead of time, we tried to kind of push you in those directions, and more writing in multiple locations through your capstone project is super welcome and super good to have those topics. Yeah, Bryce?
Using that like, in doing that, like, putting different pieces of that writing assignment throughout the paper, do you think we should just, like, not have that in the paper, like, that actual whole block of writing we did in the paper, and rather break it up and put it in places, or should we just have that and then repeat ourselves in different places? Like, refer back to it, you know what I mean?
Yeah, I guess I would say that it would be a case-by-case basis, and that you might have like an appendix that has the, say an appendix that says, this is my whole writing assignment, even though I've included portions of it. I think that would be a good idea, to kind of to have the writing assignments not as sections in the paper, but as appendices, and then try to move as much of those as you can, not by cutting and pasting, but just by copying and moving in and changing around. Yeah,
Okay.
and because a goal of a capstone project is it is not just a scientific term paper. It is a core requirement, and that's what the core requirement committee wants capstone students to be pushed on. Is, well, you're using your expertise in biology to address principles in the core general education in college, like that. Yep, and everybody's doing that, even Luke, who's not, required to do a capstone. All right, so that's a straight up push in, but it, again, it's on there for a purpose, and I think it's a really good one to talk about, to bring those two together. And in looking at the one you want to compare and contrast it to, I hope it helps you in both, both the exam and your capstone project, and then you might need to learn about that example some more, and in learning about it, I hope that helps. Okay, so here's another example. So this is about toxicology, and I wanted to start with the second sentence, begin by describing principles of traditional toxicology that we learned about in our course. But we didn't learn about this too much, and we didn't have any specific reading assignment for them. But we could flip it and say, start with thinking about what are the challenges with endocrine disruptors in traditional toxicology, okay? And this is a really good slide where I just tried to bring out some of those. And they're modern challenges, and we talked some about traditional toxicology and how traditional toxicology was really originally rooted in radiation exposure. And radiation exposures at low levels, there could be acute radiation sickness, but at low levels, the big concern of radiation is, DNA mutations. And so the traditional toxicology is really built on, I hope the toxic thing is a universal toxic, like radiation, that's going to affect every little thing because it's going to mutate its DNA. At a low level, at high levels, radiation sickness causes other kind of trouble, but at low levels, that's the deal with radiation. It's causing some random mutations like that, and the rates of mutation are going to be really similar. So we could use some simple organism and expose them to radiation and get a feeling for how dangerous this amount of radiation is, or this type of radiation like that. So that's traditional toxicology. The other thing about traditional toxicology is, radiation exposure is a super linear dose response curve. More radiation, more mutations, like that, like that. And then the next thing is, everything's the same. It doesn't matter what age you are. There's no such thing as a sensitive period of radiation, like that. There's no such thing as a species-specific thing, or there's no such thing within a population of a range of responses of individuals, but depending on their allelic composition, right, like that. And that's traditional toxicology, and I want to have you write about that, the challenge of this modern research. And so these are examples of the modern challenges, and you could just flip them around to explain what the traditional view was, right? Traditional view, we're going to do a dose response curve. Modern view is, what if they're not linear? Because the endocrine receptors are really not linear. Super weird, like that, right? So that's a good slide to look at to build that answer about compare and contrast this modern endocrine disruption with more traditional views of toxicology. And because it's very, very challenging now, and maybe I would include like described examples, because we also talked some, and our reading did about some of these endocrine disruptors have been known for a while, or thought to be an endocrine disruptor story, like the multiple limbs in frogs. They've been like studying that for almost 20 years, and they're like, we still aren't really sure exactly what's going on, So what's it, and maybe an example to include in describing the challenges of doing that. To really pin stuff down is hard to do, especially when it's in nature, like that frog story. Okay, so that's a good one. And here's another one, and I talked some about this to bring in some stuff from our first half of our course, and I talked about this idea, and this is the idea of, in our first half of our course, we ran into hormones and endocrine systems a lot, and we use that term neuroendocrine axis. And that is what the brain often is integrating environmental input and regulating the endocrine system. And so we had really good examples of that in the first half of our course, and we have really good examples of how the endocrine system gets disrupted in the second half of our course. Yeah, there could be antagonists or agonists like BPA that bind directly to the hormone receptor. But, so many examples of what has to happen upstream of that, right? You have to have detection of the stimulus to make the hormone. Then there's going to be endocrine cells in an endocrine gland that are responsible for synthesizing it, which is a biochemical process. Then there's going to be issues of synthesis and degradation, because hormone levels are always a balance of synthesis and degradation. And sometimes there are multiple hormone steps upstream. So you might have to have a first hormone that activates a second gland to produce a hormone. So just even upstream, there's a lot of stuff. And then downstream from the receptor, there are also examples of the steps that have to take place. So when we talk about this, there could be many steps that could be disrupted or altered. So it's good to make that as a comparison and contrast. And in this writing, this is a good example. For each question you should do this too, which is to try to zoom out and explain just in a general sense why hormones are so significant in the development of life on Earth, because of their just utility in regulating multiple tissues in multiple different ways, depending on the tissue's preparation for responding to that hormone. Yep. And so this goes back to that slide, where I had that slide about trying to organize our learning like that. Okay. And it's a little bit out of place though, I think, or the way I highlighted it. So we'll talk about that next probably. But that's a good question, where you could bring a lot, and it involves going back and thinking about, well, what, would you want to write from the first half of our course about an example like that? And so find an example from our first half of our course that's a good example of the endocrine system that you already learned about, and just brush up on that and be ready to combine it with, it can be two different hormone stories in two different organisms, but in comparing them and contrasting them, that's a good thing. Okay? So here's another one, and that's this one that we talked some about, and there's a good chance we would want to talk some more about that. So in each of these cases, again, we can talk about them some today, but you can think about them and look at them over the next couple of days and ask questions for about on Wednesday, or send me email about, I don't know anything about this. Okay, so let's look at some of that. So we have some examples that we covered, and again, in developing your answer, you might need to learn more and might have questions. So one is, I described these ecdysteroids. Now I see they're called phytoecdysteroids. So arthropod hormones from that are made in a plant, so phytoecdysteroids. Ecdysis is the biological term for arthropod molting. When an animal sheds its exoskeleton, that's called ecdysis, and so this is the traditional molting hormone. In insects, it evolved along with metamorphosis to have a new role in regulating growth. And so we saw the example from my own research, right, where I brought that in and I said, when chemical companies are going to sell me purified ecdysteroid, they purify it from an animal source. So that's a great example. And then in our reading, there was this example of a sheep or a livestock that graze in meadows. Those animals are sensitive to this corn woolly plant that's a weed that can grow in a meadow, and it makes this molecule called cyclopamine, and we talked about that, and it's in our reading. And so in that case, the disruption is very well known. It's this disruption of sonic hedgehog, and that's a good example from developmental biology, a very famous signaling molecule that's used over and over again and plays a big role in vertebrate face development. So the neural crest cells migrate to form the vertebrate face, and the sonic hedgehog plays a really important signaling role like that. And then example I hadn't thought of, but I realized is when we talk about arsenic, that's a metal that's actually just naturally occurring. The big source of arsenic is not as a pollutant. It's just it's leaching out of rocks, and so it would be a natural source too that you could use as a good example of an endocrine disruptor, because we learned, yeah, it's a metal, but it really disrupts those nuclear receptor family members for sure, like that. And we just briefly mentioned this one, but there are naturally occurring endocrine disruptors in plants and fungi, and maybe you learned more about that. Oh, another thing is some of your examples that you pulled on, to bring to our discussions. You already have a good example there to develop, maybe a little bit further, right, like that. So absolutely great. And then, we saw other students with good examples, right, as well. So, that's where we're getting our examples from our reading and from stuff I brought to class and stuff from students brought to class. But good examples of that idea for this question, that we can see these from natural sources. And that's, a surprise, because the field is dominated by pollutants and molecules in the built environment. So artificially synthesized molecules causing these problems. But they happen sometimes to an organism's advantage in nature as well. So that's a good one to dive into. I think I have a couple slides of that. Yeah, I just was a little bit surprised. So this cyclopamine primarily is affecting that hedgehog or sonic hedgehog. And so if you talk to an endocrinologist would say, yeah, you want to talk about an endocrine disruptor. Well, an endocrine factor is a hormone, and the definition for a hormone is that it goes throughout the whole organism, diffuses throughout the whole organism. And sonic hedgehog is not an endocrine factor. It's not a hormone. It's called a paracrine factor. It's a local signaling factor. And so it's, a little bit of a thing where they're like, yeah, well, we're going to call it an endocrine disruptor, even though it's not an endocrine factor. It's a paracrine factor. And so that's just the reality of that. And I hadn't actually said that out loud, and so I thought it was good. And then, oh man, that sonic hedgehog is so well characterized, and so it's very easy to characterize this cyclopamine and say, well, what do you think it's causing trouble with? Because we see this tremendous facial birth defect in these livestock babies. And you're like, well, let's look at some candidate molecules. And they found, because of that, they found, cyclopamine is toxicity is due to binding directly to one of the proteins in the sonic hedgehog signaling pathway. So sonic hedgehog binds to a receptor on the cell surface patch, and then they interact with a signal transduction molecule called smoothin to bring the signal into the target cell, and cyclopamine binds directly to smoothin. So just a really great example for our course of this, environmental modulation of developmental mechanisms. Because we know that developmental mechanisms really well in developmental biology. Great. Trying to remember why I highlighted that. Oh, I just found, I found some recent, this is an overview, and I found some really recent review articles on Google Scholar of these topics. If you remember in class, I said I don't know if people consider that the phytoestrogens produced by soybeans would be considered like a defense mechanism, but apparently they're broadly considered to have evolved as a naturally occurring defense mechanism to try to prevent animals from overgrazing like that. Here's a bunch of soybeans. If you eat them all, you're going to be in trouble. So don't eat us all like that. And examples of that in fungi as well. So if you pick this as an example, back to this question, right? And pick an example, and if you want to pick those, there's a lot of general public awareness of something in legumes that could be kind of a endocrine estrogen mimic, and so it's a realistic concern, and there's some really nice articles, and I just wanted to show this highlighted sentence, and so this is about metabolites in plants. On metabolic pathways, there's some molecules in plants that can be endocrine disruptors, and it says that they may and so these secondary metabolites can end up in the environment because of plant decomposition. And so it's in the plant, and as the plant's breaking down, it's got those molecules, and some of those persist longer than other molecules in nature, in another organism that's eating the decomposing material, like a scavenger, a bottom-dwelling fish, and so forth, like that. Agricultural runoff, and then new one to me is this botanical pesticides. So some of those include secondary plant metabolites, and although they're considered by home users to be a safe pesticide, apparently some of those there are some concerns. Even though they're a natural product at a high concentration, they can cause endocrine disruption like that. Okay, so again, you might use that example to dive into, do some more learning about a topic that you're like, I would like to plug this into the exam because I'm interested in it, like that. So then a little bit slightly out of place, because just a little bit out of place. So here's the next question, is the idea of these forever chemicals, and it's a good question because there's so much in the news. And so pick one of those and explain what its problem is. And in general, the concern with forever chemicals are these especially, and we would be interested in writing and learning about that first one. So there's some forever chemicals that people are scared of because they're carcinogens. Some suppress the immune system, and these issues would be in humans or in wildlife, like that. Yeah. I think the kind of the dominant concern is their endocrine disruptors. So when people worry about forever chemicals, they're worried about endocrine disruptors, even though they might not know it. They might know, oh, just these are dangerous, like that. And so it's a very good thing for us to dive into this, very topical, very modern, very challenging, right? traditional toxicology, has a hard time struggling to try to deal with these forever chemicals. And then we have principles, and this is, I think I have a slide here that just says, right, this would be the great example for you to try to write some in this answer about accumulation and biomagnification, right? So it's another example where you want to zoom out and be like, what's some of the big picture concepts and principles in our course, like that. And those are big principle ones. The environment is set up for these concerns. And for forever chemicals, well, that's just a gigantic concern, right? Is that the bioaccumulation during the lifetime of us, right? Big problem for traditional toxicology. This is not built to say, well, what about something, that's going to build up over a lifetime, like a lifetime of a mouse eating their little mouse chow, in the lab. You got to study them for a few years to really see what the bioaccumulation is, or the magnification in a food chain, right? So, big concepts in ecology that we would want to bring into the answers on our exams, like that, and then dive into the details of this. And I talked about some of these PFAS and PFOS forever chemicals briefly, but really spent more time talking about the PCBs that have been banned for a long time as being just a really tough example because they've been banned, but they're still in the environment and causing big trouble like that. Yeah. And so from PCBs, we got into dioxins, and that would be a good example to bring. But if you have learned or already knew something about PFAS, PFOS, or other, popular forever chemicals, develop that, answer for this type of question, like that, okay? And so we just go back to that, because that's another question, like that. And that's that would be a good question for the exam. And again, trying to bring in a different example of, from some of your other questions and different ideas, like that, okay? And I should be pausing and seeing if people have questions, like that. And because we're not in a hurry this week, we're doing great, and I have some more new stuff, but not stuff that we have to read new research articles about or new reading assignments, just more stuff. Yep. And then also the idea that Wednesdays are last class, and so you can also bring questions then too, okay? And that aryl hydrocarbon receptor is a really good one. The dioxin example in PCBs also fit into that. That's just a really good example to read and learn some about. You might pick something else, but that's a really good example for dioxin, especially in PCBs. So we talked some about that and about how they act and how it activates gene expression to detoxify things. And that detoxification is a very good thing for us to learn about some in our course. And then how ultimately it leads to this immunosuppression, like that. Great.
How does that part fit into this last question?
Yeah, so that dioxin is a good example of a forever chemical. Yeah, and the question is, pick one or more, and so, find a list of forever chemicals, and there's PFAS and PFOS, and I don't know what the other ones are that are in the news a lot, that are in like non-stick Teflon type coated cookware and so forth. But I said let's look at PCBs and then dioxins, and because they're just such a good example. And it's such a good example because you want to talk about a forever chemical. Well, they've been in the environment since I was a kid, right? How much of dioxin do you think I have in my body? Up until third grade, one of my chores was to go out and burn the trash every week. There was no trash pickup, and you would haul big stuff like car tires to the dump or a washing machine to the dump, and everything else we just burned in the backyard in town, you know. And I played air guitar in the smoke once a week. And I'd come in and my mom would be, "Hey, you stink," like that. And I'd be like, "I'm a rock star, Lois." Yeah, literally. I mean, one of the top 10 highlights of my life is that most people in my town burned their trash in an old, like a 50-gallon or whatever those are, not 50, bigger, oil drum. You know, just a big metal drum, dump it in there and burn it. And the highlight for me was we had like a wire mesh trash space, so I can see everything burning. I'll see that styrofoam melting and all the colors coming off newsprint and crap like that, and just be right up there with it, you know, just going like, yeah. My mom super loved me, but she never would have thought that there was some danger in going out and burning the trash, you know? So yeah, so that's just a good example. At least it resonates for me, you know, because I'm like, oh my God, you know, like that, you know, like just must be Wackadox and like Davox, you know, like that, right? And so I stuck with that. And also this aryl hydrocarbon receptor is a famous example. It's not considered, you know, normally considered a hormone receptor. So that would be a good example. That's how that fits in there, is to pick one and be ready to write about it like this example. And bring up, you know, bioaccumulation, biomechanics, oh my goodness, you know, like that. And that these are part of the built environment and then part of pollutants, right? I think dioxin is more considered a pollutant rather than, you know, something that people try to manufacture and sell. They sell other stuff, and when it breaks down some or gets burned, when those molecules get burned, they can break down to dioxin. Yep. And, you know, bioaccumulation, biomechanics, it's just, they're organic molecules that are not natural in nature. So organisms didn't evolve the ability to catalyze their decomposition and detoxify them by decomposing them. So the aryl hydrocarbon receptor is a great example too, because its big deal is it's going to activate expression of enzymes in cells, especially the liver, to try to break down whatever molecules this is. You know, I'm going to try to break it down, whatever it is. And so the aryl hydrocarbon receptor is significant in some of these other stories we have as well, because the body's like, oh, I'm going to try to break this down. Like that. Like, you know, those phytoestrogens, the body's trying to break those down. And one of the issues is they bioaccumulate. Well, they bioaccumulate if you can't break them down. You know, so it's a good example. I'm happy for any example, right? It's not like, you know, I'm like, I wanted to read a lot more about Dave Oxon on, you know, on your exam, Lou. You know, pretty, you know, like, I don't know, not that psyched, you know, right? It could be anything. And it could be, you know, some other example, you know, that you know, dive into. Because I think in preparing these examples, you know, it would take you half an hour to kind of pull notes together and be like, I could write about that, you know. And so that's a good question. And because it brings up good specific molecular stuff and good broader principles. That's what we're trying to think of, like that. And in thinking of other questions, you know, that you might have been learning about, that you'd like, I'd like to plug this in, and I don't really see a place to plug it in yet. That'd be great. All right.
It's okay that if we use, like, because it said like one or more, but so we can use, even though they're connected, we can use both PCBs and dioxins together.
Yeah, and I say that on several of the questions. I say use one or more, right? Because sometimes you want, you know a piece of it, and it's like, they're really good for this part, to really bring up these principles, but then I would like to switch over to this other one, like that. It's not good to have that be a mile wide and inch deep, where you just list as many as you can, right? Forever chemicals, I know that I'm going to say 8, you know, and I'm done. right? So listing more than one is just gives you the opportunity to not say, oh, there's a piece that this other one brings that I would like to write about, because you know, I kind of need to combine them to get all to be done. That's great. Yep. Yep. All right. And so then we're doing great. There's that one. I wanted to actually make it as a question. You're super welcome to send me a suggested question. And you should try to do it by Wednesday, because that'll be the last time I see you. And if you send me one over the weekend, I'll be like, yeah, I'll just put this in, right? That's hard to do if the other students have no clue where that's coming from, right? But that's possible. And then this is the last suggested question I have, but it's also one that we can spend time talking about today and Wednesday. And so it's about this idea of evolution that we dealt with a little bit. We ran into it a couple times, because it was in our first half of our course is where this is particularly relevant. And it has to do with the idea that when there is phenotypic plasticity based on environmental factors, that means the population of a species has variety, and that variety can be selected for in natural selection. And so it seems like this phenotypic plasticity could be a mechanism that would lead to evolutionary change. And we saw Bryce and Emily each had examples where they're like, this paper they brought to a discussion was about the idea that maybe the plasticity of the frogs, or in Bryce's case, or in Emily's case, this little fish, could have been the mechanism. So we saw some phenotypic plasticity in some closely related species, and then there's a species that lacks the phenotypic plasticity, but it seems to be lacking it because it's living in an extreme environment, and so it's gone with just that one form, and not even maintaining the plasticity anymore. It's just going to be living that form. So that's what we're going to want to talk some more about here, and so the question would be to bring those examples up and see, and you're not going to have to write about this, right? So, right, there'll probably be a question about the exam, something like this, right? And you're going to have options to write about, but you're going to have to write about most of the questions. So, if you decide not to. But just to go back to Bryce's question about dioxin, there's not going to be like a fill-in-the-blank question where you have to know the word dioxin or the aryl hydrocarbon receptor. You just have to know a lot of good stuff, but you're not going to be directly called out the way you are in most courses on, well, what's dioxin? Yeah, so you're picking and choosing, and so the same is true here, where you can start thinking about picking and choosing about this topic. And to talk about this topic, what scientists tend to do is they'll pick an example, like we've seen, or other example that you find, but they also talk a lot about just normal developmental mechanisms. So that's what I have some more of for today, is to talk about some normal developmental mechanisms, to put it in the context of when we say phenotypic plasticity, what are we talking about? And when we're talking about that, how could that evolve? Yep.
Is that how you would connect the examples? So to answer this question, you can explain let's say the frog example, and how they're very drastic, extreme environment. There's not as much or little plasticity, and then I'm kind of struggling to see how you get to that second part of the question.
Yeah. So it's true that that I would say that's a thing that is hard. It is a struggling thing, is to make that connection between the two, I think we should talk about some of this today and Wednesday, and also toward the end, as we get into that, also say, okay, now can we see how to make those connections some? Yeah. Yeah, because the slides I have are kind of a gap like that. But again, the idea is there are examples we could find in nature that we're like, oh, it looks like evolution has taken advantage of phenotypic plasticity based on the phenotypic plasticity of closely related species, and then the lack of it in this species, but yet it has, it's just one of the range of plasticities that its near relatives have. Yep. Yeah, and so this is the last example question I have today. I said the exam will maybe have 10 questions, something like that, and you'll have to write about 5, right? Like 5 20-point questions. Similar, hopes that you'll write, 20 points worth of questions on each one like that, yeah? And so, maybe I'll have 1 or 2 more like that, and maybe the wording will be changed around a little bit, like I said, one of them anyway, I said, oh yeah, you should have examples, like that, right? And these questions are not like fill in the blank questions either, where you're just like, you have to have all these, there's a lot of flexibility, but I'm giving you some guidance to see what you should be getting ready to bring to the exam. Okay, great, all right? And so, I'll start talking about about this idea of how does evolution occur, and developmental changes occur during evolution, with examples of that, and they won't be examples of phenotypic plasticity, but rather they'll be good examples of development and how development can evolve, and then that would be how we could get to evolution of phenotypic plasticity and changes like that. Yep. So yeah, like Bryce talked about, there's kind of a gap between those two because of this is a new field. Yep. But we should pause there too, because we kind of rolled through this part, and now we'll go back to talking some about what's this, what's that. Yep. So other questions or issues people have about anything at all? All right, so let's look at some of this stuff, and this is going to be, all this stuff is going to fit in that context of evolution of developmental mechanisms, and then ecological developmental biology, and how those could evolve. Yep.
If we're gonna make a question to send in to you, should that be focused on this half of the course, like the second?
It could be anything.
Okay.
Yep. Yeah. Oh, I would say it'd probably be good to have it on the second half.
Yeah.
Because I said there will be a little bit from the first half, and I already suggested a couple questions from that were leaning more toward the first half.
Yeah.
One of them was compare the first half and the second half with the endocrine factors, and so we probably. And I think the other one is, write about your capstone, and I think everybody except Madison is doing capstones that are more about our first half than our second half. Like that, yep. So yeah, if you're to send me questions, it'd probably be better to focus on the second half. Yep. Other questions people have about anything? All right, so let's look at some of this. And just to bring up these terms, and so there is the older term is this term evo-devo, evolution of developmental mechanisms, and there's just a lot to say about that topic. In the last 50 years, when biologists discovered great details of molecular mechanisms in development, all along the way, it's been very clear that these have tremendous significance for our understanding of evolution. So we'll talk about those developmental mechanisms some, and then hope to connect them to the topics where some people like to call our course eco-devo, ecological developmental biology, and it's because it's mirroring that the powerful evo-devo term, and that is the the bringing together of what, were seemingly different topics, and they get pulled together because of the research and the discovery and the biological principles shared between them. And so that's what we're talking about. And so this is just a article, or the author of the book that I've used in this course wrote about how evolution occurs. And so here are some unusual terms. There's a good chance you've never heard these 4 terms before, and so I don't think you need to try to remember them. But here's what you can remember, and that is that evolution is a change in allele frequency of a gene in a population. And so that, what I just said, brings together genetics and evolution. And 100 years ago, genetics was a new science, and as it emerged as a new science, it became clear that there was a big relationship between genetics and evolution. And so people still refer to that as the modern synthesis. People are starting to appreciate that the evolution by natural selection took place by some change in genes. They didn't know what a gene was yet physically, but some change in genes, and ultimately change in the allele frequency of those genes in a population. Like that, okay? And so this is about that the sort of the basic unit of evolutionary change is the gene, and then this is the 4 ways that a gene could change. So there are only 4 ways that evolution can occur. And these are the 4 ways. And they're just the 4 ways that a gene could change. And so you can think about that, because it's surprising to think about that you'd never have heard these terms, but this is the only way evolution occurs. And maybe there's some exception to that, but it would be an exception. And so what are they? So we can just slide off here and just do this, because they are really obscure terms, the old-time genetic terms, but they're built on just a very modern and very straightforward understanding of the organization of a gene. And so this is going to be DNA, and my drawing tab I had to replace during spring break or sometime, and it's weird because see how it only draws over to here and here? So I only have that much drawing surface, and it only happens in this classroom even when I'm not plugged into the projector yet. So I don't know what's going on. I'm like, I haven't solved it, and it's like, this is where it's a problem, and I don't know. And it's not always been a problem, it just sort of showed up. So I gotta draw this little space. But this is DNA, and so we could talk about a definition of a gene, and a gene is physical, we know that now, and those terms came before the physicality of gene, and it's made of DNA, composed of DNA. And then the next thing is that all cells on Earth contain DNA, as the primary inheritor molecule. Some cells have thrown away their DNA, stuff like that, like human red blood cells. But as the primary inheritor molecule, all cells have it, and the structure is the same. And then there were these 3 really great French molecular biologists in the late 1950s. The most famous of 2 are Jacob and Mond, and they discovered this in bacteria, a gene called the lac operon, and they won a Nobel Prize in about 1962. So less than 10 years after the discovery of the structure of DNA, these scientists won a Nobel Prize for understanding how DNA was organized. And the key way to describe what they were discovered is that they thought it was going to be the case, they predicted it would be the case, and it turns out to be true. And that is that, DNA is the is the primary heritable molecule, and that we have all that information we hear from our parents, and like a computer where you don't open all the files at once, that's what DNA is like, is that you output portions of it. And so the gene is just a unit of information output. And it's based on that you're not gonna use all the information, you're just gonna use a portion of it. And the portion that gets output gets transcribed into RNA. And I think the only way to output information that DNA is holding is through transcription into RNA. And so that becomes part of the definition of gene, is that gene is physical, made of DNA, and it's composed of two parts. And one part is going to be the part that's got the output information, and the other part's going to be the part that's the regulation of the output. And that's a gene. It's made of these two parts. Ooh, that was the wrong button. And even though that microbiologists, like those French scientists, they use different terms, it's just a universal definition of a gene, is it's composed of these two parts. And one part is the transcription unit, the part that's going to be output. And so that's the transcription unit. And the other part we'll just call, it's a good name for it, because people use some different names, which is frustrating for students, is the regulatory region. And so with that, we can say there are only really two ways a gene can change. You're going to either have a mutation occur that's in the transcription unit or the regulatory region. And you can either change what gets made or its output. And that's where the four terms fit in. They're just four ways to change the gene, and they go like this. One is the mutation could be in the transcription unit, and you will make an altered product. And with any mutation, you would just ask about it, like sickle cell anemia. A person who has sickle cell anemia makes an altered hemoglobin, and the hemoglobin molecules bind together and form polymers that change the shape of the red blood cell. Where's the mutation for that? Well, it'd be in the transcription unit. To change the product, the mutation has to be in the transcription unit. And then over here, we can have mutations in the regulatory region, because the regulatory region is physical, and there are really three things the regulatory region controls, and they are where, when, and how much of a gene gets made. And yeah, I think that, one of the, like in science education, there are always these weaknesses. And one of the weaknesses in science education is in intro biology, you learn a lot about the product of the gene. Like students are like, yeah, it's going to make a protein. But not even all genes are coding proteins. Many of them are non-coding proteins. And so students are like, wait, they're forever kind of stuck on that. What's this make? And you're like, well, it doesn't make a protein, it just makes RNA. But yeah, there's that category, is you could change the product or you could change the regulation. And the four ways to change a gene are change the product or change where, when, or how much. And those are three distinct things like that. And we can give an example of that. And so a good example is that animals have genomic equivalents. So in our bodies, we have the insulin gene in every one of our cells. And the implication with genomic equivalence is that, oh, it looks like DNA for most organisms, it's just easy to copy DNA and do a good job copying it. So it's like a modern computer where you can just have tons of memory. Memory's cheap, and similarly, DNA is cheap. In bacteria and some single-celled eukaryote, there's natural selection pressure to make the genome small, but there doesn't seem to be really any natural selection pressure. So there's literally a lot of junk DNA in most eukaryotes because there's just not a lot of natural selection pressure because it's easy to make it like that. So it's easy to have a lot of DNA, and so it's easy to have genomic equivalence. And so similar to having insulin in every one of our genes, we have hemoglobin in every one of our cells, hemoglobin in every one of our cells. And we also, all vertebrates and all animals have that have muscles, we have all the muscles to make muscles in any cell. We're just not making them. And so developmental biologists are interested in the idea that, genomic equivalence opens the door for a lot of potential evolutionary change. Because if we activate the gene expression in a new place, time, or amount, that could be a surprise like that. And then we can find examples where there must have been that type of evolutionary change during the evolution of it. And so a good example would be muscles in the nose of an elephant, because the elephant's trunk is its nose. And ancestrally, 12 million years ago, there were no muscles in an elephant's nose. We don't have any muscles in our nose. People who can twitch their nose don't have muscles in their nose. But we have the genes to make muscles and all the genes to regulate the production of muscles. And so it really looks like evolutionary change it includes a lot of changes in development, evolution of developmental mechanism. Just start making muscles in your nose, and that doesn't make them automatically have a great trunk. It still takes millions of years to evolve, but you open up this just really obvious door toward evolutionary change like that, right? And in that evolution, it's not the muscle proteins that changed, it would have been the regulatory region like that. And so it gets complicated because the regulatory regions are regulated by proteins, and those proteins are transcription factors. They themselves have genes. So we didn't change the muscles, but we either changed the regulatory regions of muscle genes, or we could have changed the transcription unit of the transcription factors and changed them. So it gets complicated a little bit, not terribly, but a lot of the story is here like that. And so we can just go back to those terms, and you can see that even though they have odd words, and you probably will never hear them again, see, they say change in kind. That's that first example. You're going to make a different product, like a different hemoglobin protein with sickle cell anemia. So that means the mutation has to physically be in the transcription unit and in the coding region of the transcription unit. And then these are just where, when, and how much. Regulatory region changes like that, and that's it. And so there's some great examples of that. And so where the gap is we'll talk more about some of these examples, and the examples are not for phenotypic plasticity. They're just great examples to illustrate, like I just did with elephant noses, like that. Illustrate these types of mutations that are thought to have occurred, leading to some evolutionary change. And that's Evo-Devo stuff. It's not Eco-Devo. So we have to sort of set the stage with Evo-Devo to be able to kind of mentally and then practically get to, phenotypic plasticity. But we have built the island, we're building the island of where the Evo-Devo and the mechanisms of development occur, and then we built the island of phenotypic plasticity because we talked about mechanisms of the plasticity, right? Dive in and say, okay, the ants are feeding the queen larvae this royal jelly. and that's gonna cause changes in this gene expression, and so there's gonna be plasticity there. So the mutations and evolution would occur in the genes that are involved in the plasticity like that. And I think, just a couple minutes to go, but this is probably a good time to pause, so you get a feeling that I can talk about this some, and it's a little different topic, but it brings in some good part of our course that's advanced, but it already came up a couple of times. And there could be a final exam question on what we're talking about, and you might decide, yeah, this is cool stuff, I want to dive into it some more and be ready to answer that question as one of my 5, like that, okay? So that's where we're at, all right? So a couple minutes left, but I think good place to stop. Does anybody have any questions or issues right now about stuff? Yeah?
So are we gonna, next class, are we gonna do more like going over like questions stuff, or do we have any material?
So I said, I gave 9 possible questions today that I just, made up in the last few days like that. They're good questions, I made them up. They could be on the exam, so, probably will be, and maybe 1 or 2 others, depending, like that we got to talk about on Wednesday a little bit if we come up with them like that, You could also say, I think you should delete, that one of those questions because I hate that topic, like that, and that would be fine, just to replace it, you could say add my question and delete that other one. That would be fine. But so yeah, just be a little bit more on, here's another possible question. Wednesday would be great if you process some of the question things and wanted to talk some about it, to say, well, can you describe that aryl hydrocarbon receptor again? Like that? That would be great. So to reflect on that some for Wednesday, and also, I need more questions, but especially what are we looking at and what help do you want to get from class, like that would be really good. And then, the other thing is I'll just drive down into this some, which would be limited to 1 question option in the final, and just good learning, good additional learning in this field. Yep, other questions or issues? I used to say, does anybody have any questions or comments? And I kind of stopped doing that because there was a student, Haley Defner, named Haley because she's the greatest human being ever, and Haley would often want to stand up to make a comment. She'd be like, I have a comment. I'd be like, all right, and she'd stand up, make a comment. Yeah, just a really great character, real scientist kind of person. Yeah. Oh my goodness, yes. But so questions, issues, comments. Yep, but then we'll call it for now, and we'll pick it up again on Wednesday for our last class meeting. Woo! And then our finals